Okay, hey everyone. So as your request, yeah, I'm trying to make a video regarding about the time to tutorials, the moving pictures using the command line interfaces, yeah, using the Ubuntu. So I hope this could give you a little bit of a sharing of the light, yeah, of what how we do the metagenomics analysis projects and also how to analyze them as well. Well, Galaxy do have their own merits, yeah, for you that maybe do not have any experience yet, or maybe do not want to go for the command line interfaces, then just go to the Galaxy. Well, it is a web server again, and sometimes it does need some kind of maintenance, and you also have a very limited space. Yeah, 100 gigs it seems too much, yeah, but again, if you do a lot of data, if you have a lot of analysis before that, then 100 gigs is going to be poof away very, very fast. So one way to do that is actually using the command lines. Yeah, it doesn't hurt to learn new skills, but again, I'll give you this for request. Yeah. So let's see a little bit. So I have my Ubuntu already running in here. And then this is with assumption that you already installed the Qiime 2, yeah? You already installed the Qiime 2 so that you already have that up and running in your machine. So let's see, check if my mine is already there. So actually I did a little bit of that. And then to check that is that I can use conda env list to see the whether if my Qiime 2 is already installed in the environment. So if you already install it per the instruction, then you'll have one environment called Qiime 2 2021.8. Yeah, this is the latest one as the usual. So and then I create a folder called Metagenomics in here just to make it a little bit tidier. So we don't really have to the data is not going to anywhere. Yeah. So to do that, let's see what's inside. Actually, I already make the Qiime 2 moving pictures to moving pictures. This is my testing. So because based on the tutorial here that I opened, yeah, I need to make a new directory. Yeah. So of course, in here, if you open it up, there is here that you can just copy paste the comment itself so that you can work it a little bit faster. But then let's look a little bit of the idea, right? So first, let's look at the creating this particular folder using the command here. Yeah, just click that and then to paste, yeah, to paste is not using Control-C or Control-V, but Control-Shift-V or Control-Shift-C to copy. If you just Control-C, then it will actually close or end or shut down every single that comment that you are removing in the terminal itself. So to copy and paste, it needs some fee, yeah. So because I already put that, yeah. So there you go. Yeah, Control Shift V because it is actually two commands. Yeah, this one command and then this is the command to change the directory. Then we have this folder already. Yeah, Metagenomics Qiime 2 moving pictures tutorial. And then after that, we will need to take the sample metadata, right? So you can use either download it straight from the browser. So this is the link that you can just click and it actually will prompt you to download it by the browser. Or you can use the command line entirely using either wget or curl. Both command actually do almost the same thing. Is that how to do it is a little bit different though, yeah. So we can go wget or curl, be my guess. So I will just use the call wget. Control CV again. This is still in the directory of this, yeah. So don't do it. Okay, so yeah, after you type in enter, then it will do what it's just told, yeah. So the metadata TSV, yeah, is going to be inside the folder. So let's see a little bit. There you go, sample minus metadata.tsv, or a tab separated value, which is usually the one that you use for Excel. Yeah? Okay, so we have this metadata, and of course you can check the other, yeah, Qiime for validating the sample metadata in the case you want to learn about the metadata again. Then of course, Qiime2 also have some tutorial to format your metadata. I suggest you to check it if you want to go deeper into the tutorial, but for now we are not going to do that here. Okay, and then here we need to obtain and import some kind of data, and then you need to put them into a single folder here. So just to make sure that it do not spilling anywhere. Yeah, Kadir EMP single end sequences. So this one will create the folder EMP single end sequences. Then after that, we will need to download the FastQKZ, yeah, FastQKZ to check for the data. Yeah. So actually we have both here. So let's just do both. Okay, this is going to be related to your speed, yeah. I'm in campus, so this. Okay, so that's the first one, and then this is the second one. Yeah, we get the investment first because I thought this is a similar. I don't know. Oh yeah, this one is for the barcodes. This one is for the sequences. So both need to be downloaded so that you can work on the next part. Yeah. So barcodes and sequences. Yeah. So remember during the NGS analysis. Yeah. It is in NGS usually have some kind of barcode to differentiate between one sample to another. So this is pretty common practice in metagenomics analysis as well. Okay, so our data is already there. So based on the command here is actually duplicate. So this forward slash is just to make sure that you can put the command in one line below them. So it is just only for quality of life. Yeah. So of course you can just widget and then -o. This is the output directory, and then after that space, put the link to where you want to get the data. Yeah. So pay attention that here when referring to the place, to the output of the command, and also from the websites that they use a double apostrophe. Yeah, this is usually required for the WK. Yeah. So here the command is actually to put the output in the folder that we just made earlier, EMP single and sequences, and then you put the sequences in there. Yeah, so this is the file that you want to WK to write them in from this particular data. Of course, you can use another name. Then the result will be that particular name. Yeah. Okay, so that's why when I click the ls here, nothing in the folder of Qiime2 moving pixels tutorials, because everything are in the EMP single and sequences directory. So maybe let's just look at it. Yeah, barcodes and then sequences of FastQ.kz. Okay, so we already get the data. So next we will need to do the Qiime2 semantic type. Yeah, sorry, not semantic type. So it's kind of like an artifact and then import some kind of sequences. Yeah, so now we are going to use the Qiime. So this is a good starting point to finally putting up your environment. Yeah, so here, look at this. This is still base, so you need to activate the environment first. Yeah, so the last one, because I just downloaded data, making a directory on something like that, I can just use the base. but because the next step needs to use the tools that coming from the Qiime, then I will need to activate the environment. Yeah. So to activate that, conda space activate space the name of the environment. Let's see again. I forget the name. Time to minus 2021 and dot 8. Yeah. Time to again. I forget again. 21.8. Sorry. Oh. Oh yeah. Conda active. Yeah. There you go. Now my environment of time to 2021.8 is already uploaded and active and running. So then we can use the tools from the Qiime. So next, this is to import some kind of data and barcode associated for each sequences, and then we will look for the import. Yeah. So just copy paste this if you don't want to typing a lot of things. So just that magic happen. Yeah. So we have imported EMP single sequences as EMP single ND format to EMP single N sequences.kza. So kza is an artifact that used to Qiime to check for certain kind of data. We can see the inside of the kza and then using the Qiime tools pick. Yeah. Yeah, so we can use the UID, the type, and the data format. Yeah. So keep a note of this one to see for the artifact and yeah. Right. So we already get all of the data that we need. We already have the barcodes. We already have the sequences. Then next up, we will do the multiplexing. Again, if you will need to look for the sample metadata again. Why do we need to do the multiplexing? Yeah, which is usually done because we have a lot of samples and we just have one machine. Just for the efficiency, you just want to run every sample that you have in just one run from those one machine only. So then you're usually adding the barcode. So then we will need to demultiplex them and then sorted them out based on the barcode, right? So then they can be in their intended place to be. So for the multiplexing itself, the Qiime already have their own tools called tmux, yeah, tmux. So this is how they do it. So the input is the AMP single N sequences TZA. The barcode is from the one that we take from the .tsv barcodes, and then also the column barcode sequence that we also did downloaded as well. And then the tmux details and tmux details, the KSA is for the output, the per sample sequences, and then error collection details. So we need the single N sequences, the KSA, and then the sample metadata, and then column barcode sequences. So I think the sample metadata is not yet in our directory yet. So yeah, it is still on the this one. Yeah, maybe let's just try to run it from the outside. Let's see what happens. So sometimes, yeah, for putting the input or anything, make sure that you also know where the data is. Yeah, so they need to be know where they are, the data will be. Yeah. So then again, it's actually running because they look, yeah, the MP single encode sequences of KSA is in our working directory. The sample data TSV is also in our working directory. Yep. So that's why it works. Yeah. Right. So that's for the demultiplexing. Yeah, pretty easy. Yeah, as long as you know the sample metadata, and then you already know the backup sequences that you use for your sample. And after that, just run the tools. Yeah, it will be creating these two artifacts. Yeah, the demux details and demux.kza. So then we want to just to see by summarize and then create a visualization. So then we can see a little bit more human readable. Yeah. So just run this and then just wait. Right, so now we have the demux.kzv. This is the, what they call it, the visualization format, yeah, in the Qiime tool. So to see this visualization, yeah, you can use this particular command, Qiime tools view. Qiime space tools space view, and then the file that you want to visualize. Yeah, it will prompt you to open a web browser from there. Yeah, and then they will see the result from there. So we are the multiplex sequence count, then we have this forward list of the minimum number of the 1,854. The median is that, the mean is that, the maximum rate is 18,000. Yeah, and then okay. There is no reverse rate. Yeah, there is no reverse rate, so that's why we only found the forward one. And then the number of samples is around this much with this number of sequences. Yeah, we found like 13 samples. Yeah, with this number of the sequences. Yeah. Then this is for each of the ID and the number of this forward sequence count. And of course, this is the interactive quality plot. Yeah, for yeah, it's pretty much the same like that you use a fast QC. So you see that this is the parametric 7 numbers summary, yeah, for the quality, yeah, of each of the rates. Okay, so this is still we just get the raw data, and then based on the barcode we sort them out based on the grouping, yeah, based on the grouping, based on the this one, yeah, based on the sample ID as well. So we didn't do any quality control yet, so that's why you can see that the quality is pretty bad. Yeah, right. So next up from the tutorial, we were asked to quality control and then feature table. Yeah, so creating this time will be creating a lot of artifacts, yeah, and then some kind of a name. You can actually rename it to create the decrease confusion, right? But anyway, this is one of the imperative tasks as well. So here, based on the tutorial, you will need to, you have two options here, data to or the blur, yeah. Well, based on that, you can choose whatever you want, yeah, and then both will work on the same feature though. Yeah, the idea of the tool itself is going to be the same. Yeah, so this is going to be detecting and correcting for element applicant sequence data, and then trim for the part ones. Yeah, so trim length and trim left is actually for checking, yeah, for which one of the, based on the table that you see that I closed. Yeah, so you can see that the quality, yeah, drop off after read position 120. So then we can try to delete the one that paired rates. Yeah, again, quality control is basically looking for the sweet spot of how many samples that we can delete because of the paired rates, or whichever. If you have a good rate, then you are very good to go. Yeah. Right. So let's just copy paste this particular comment because they are really, really nice to provide the comment already. Yeah, so we don't really have to type in a lot. So, okay, so here's the thing, yeah. One thing that in the command line, sometimes they will freeze out something like this because the previous command actually open up a new window from in the other software, in the other apps, yeah. So to do this is actually, you can see that press key Q or Control C or Control T to quit, yeah. So then if the tools, if the terminal is getting to be here like this, and then you see this blinking again, so that's, you know, that it is ready to go to enter some kind of command. Yeah, of course there are other way to manipulate this, yeah, to make sure that you can still use the terminal even though the particular command actually prompt you to create another kind of windows that you can use what we call the ampersand. So, yeah, but particularly doesn't work using the file. So okay, so doesn't work. And it stop, yeah, for the debug keys. So anyway, still coming back to here again, it works. So now let's see, enter comment to work on the data. Yeah, this actually takes a little bit longer, but I did check it's not that long. So at least in my copy. Yeah, you just need to wait. Well, we are pretty good, yeah, because it is actually a subset of data. Then if your data is much more higher than this, or much more FER, or much more abundant, yeah, then we need to wait a little bit. Yeah, okay, so after like a few minutes, yeah, actually it's done creating 3 artifacts. Then you can see the inside of artifacts by using the pick command or by looking at this view. Because they are really, really generous, yeah, because they already put the result here, but we are not going to do that, yeah. But here we can create a visualization about the metadata tabulation, yeah. So what kind of metadata that are still retained after the quality control process. So let's look up. And after that, we will see what's the inside of the yeah, time tools view. The name is Tuts. The queues of view. Okay. So you can see that this is the sorting, the quality control based on the sample ID. And then we can see how many input, yeah, that's coming from each of the sample ID, how many are they filtered, yeah, how many are the based on the percentage, yeah. So some actually a lot of data are being passed out, yeah, 83%. Some are really, really low, like 40%, 47%. Well, this is could be, I can't really say how much the normal one, the higher number that pass the filters is better. So that's when you have a lot more data to work on. But if it is just too low, like only like 10% or something like that, or the number that being filtered is very, very low as well, I suggest you maybe think about resampling and something. Yeah, and then it's also looking for the noise, if there's some kind of noise sequences based on the quality control, and then the one that non-symmetric. Yeah, I believe you already know what the symmetric sequences is from the previous Galaxy tutorial. It's about the what's it called? Yeah, sequences from different kind of species or different kind of samples joined up together. Yeah. So this is also a service from the quality control as well. And then input also that is non-numeric is this much of the percentage. Yeah, so that is basically based on the sample. Okay. Okay, so that's next what's called, and then if you want to continue terrorism with the table, opposite deploy feature table next option to run this flow command. Okay, let's do that. Getting website data and table data. Yeah, it's actually just moving around, so it's okay. Okay, so we don't use the deblur. If you want to do it by yourself, be my guest. The results should be similar. Should be similar, and we can use that for the feature table and feature data summaries. So after quality filtering and then deleting all the Qiime sequence, then we trying to create exploring the resulting data, right? So we can create a summarize from the data, or maybe looking for some summary statistic, histogram, distribution, and something like that. The time, or they have the tools for that called the feature table. So first one is to summarize the data that pass the filtering process. And also the second one is to tabulate sequences to check for some kind of mapping to sequences, maybe looking for a link to plus each of the sequences. So then we can have a little bit of annotation on the particular area. So let's try this. Copy the command and then again, because this is to command, you will see this. Okay, so we have to already table.keyzv and reference the sequence of keyzv. So let's look one by one. Call it from here. Okay, so from here we have a table of summary based on the feature table. So we have 34 sample here with 770 features with a total frequency of this much. The frequency per sample, and then for all of the sample routed as well. This is frequency per feature, frequency per sample, and then, because we have 34 sample and 770 features. This one you can check for is it the barcode. You can change it based on the body size. We have example from the gut, example from left palm, 9, and both 9 from right palm and thumb. Okay, I believe the year is some sample are written 2008. This one is 2017. Subject, subject 1 and subject 2. Of course, you will need to check the what's the difference between subject 1 and subject 2. So some from the metadata they do not use any antibiotic and also using antibiotic. First that's one. Okay. So then maybe let's look for the rep 6. Okay. Course, you can check for the 7-number summary. Sequence length. Okay, you can see that this is minimum length of 120 and maximum 120 as well, because we trunk the sample to 120. And then delete everything after that, because we know that the quality after 120 is really bad. Okay, and then this is the sequence, for each of the feature ID. And this actually can click it, and then it's go directly to the class, to see what kind of sequence is that. So anyway, but we are not going to do that. First the feature, that's pretty much it. So Right. So that's about the summary and also the feature table for each the sequence that come into for each of the feature that we have associated to that. Yeah. And then of course, if you want to go plus by yourself, yeah, just click that. It will directly go there. But we'll go for the next analysis. Yeah. First one based on the total is creating a phylogenetic diversity analysis. Yeah, because we're taking for different kind of sample, yeah, different kind of places, yeah, we can actually analyze the differences between one sample to another kind of sample. Yeah, so this one using MAFT, yeah, to do multiple sequence alignment. I believe you should remember during our phylogenetic tree analysis back in method of bioinformatics. Yeah, the first one before you get the phylogenetic tree, of course, always multiple sequence alignment. Yeah, there are a lot of ways to do multiple sequence alignment. You can use MAFT, you can use the coffee. I believe I taught you how to use the coffee. In the Mega is actually access the crystal W and also muscle. Yeah, there are a lot of algorithms. Yeah, but here in the time two, they have the MAFT. Yeah. Okay, so this one is also creating some kind of tree as well. Yeah, just copy paste this. Yeah, and then just let it do the magic. Yeah, most of the cases of working in this kind is repeat. And okay, so that's the output, yeah, for the tree, and we are actually going to use that for the next step. Yeah. And then for based on the tutorial, after we create the tree, anyway, we're going to do alpha and beta diversity analysis. Yeah. Again, I believe I a little bit tells you about that in bioinformatics and biodiversity. Yeah. So we have alpha using some kind of indices. Yeah. I believe you should already familiar with this one. Shannon's diversity, and then there's also another index called FATES. Qualitative measure of community richness that incorporate phylogenetic relationship between the features. So it is a little bit complex, yeah. Then Shannon's based on the richness. This one is based on phylogenetic relationship. So that's why we need to create this for the FATES phylogenetic diversity. Then evenness, yeah, or Peel's evenness, yeah. Usually in tandem, yeah. Shannon's and FATES. Now for the beta, so based on the community, so it's for alpha is just for one place. We in the tutorial using Jakarta distance and Curtis, and then we have this unifrac distance for both unmixed and weighted. Yeah. So let's create the core matrix phylogenetic first, based on our tree that we created on the previous steps. Yeah. And then getting the core metric results. Yeah, so just copy paste this, then let the magic do the work. Yeah, it will create a lot. It does create a lot, and then it does create several visualization. Four to be exact. Unifrac Emperor, Vector Unifrac Emperor, Chakart using Emperor Grab, and also break Curtis Emperor. Yeah. So we will work on that a little bit later though. So of course you want to check, yeah, again using the Qiime tools to view and see the results. Yeah. So about the diversity matrices. Yeah, maybe let's look a little bit. Maybe let's look a little bit. Maybe for the this in the in the core metric result. Yeah. So just look, yeah, for this one the visualization are not in here, so that's why I cannot call them. Yeah, so it will be in this particular area. Yeah, we will come back to that later. Yeah. So starting of it, I kind of want to see. And this view, so how to do this is formatted result and checkout and yeah, so this is the comparison between committee. Yeah, from here you can click this to scatter based on what body side. Yeah, you can see the some kind of sample are in the double body sides. Yeah, this one is from left and right palm. This one is the cut also from a little bit on the right palm. This one the tongue and the left palm. Then for it's different kind of day. Subject. So not a lot of differences in here. And yeah, try to work around here. See, this is three-dimensional by the way. Yeah. So you can see the difference of the clustering. Yeah, the difference of the clustering. So this is something that you could look as well to see the differences between one subject to another subject, or one variable to one variable to another variable. Yeah. So again, the next I'll let you to look it by yourself. Okay. So after we already did the core matrix, yeah, the core matrix results, then we kind of look for the next step will be alpha group significance, and for using faith and evenness. Yeah, so now we can see the based on the index, so we can compare between one to another. Yeah, so this one using faith and evenness. Yeah. Okay, just wait. So then we have this automatic results for the faith between group and the evenness between group. So maybe let's look. Let's take a peek a little bit. Maybe look for the evenness first now, because that's at least the one that we familiar with. Yeah. So here, and okay, let me. Yeah. So this is for the alpha diversity based on the body size. You can see that how significant between each of those, and then the main median number for each of the body size. Yeah, you can change this to subject. So you can see from here, not really that much of difference. Yeah, both are actually in the same clustering. Yeah, should be. Then for the reported undivided usage, so for the non one, it is pretty wild. Yeah, pretty wild one. While the yes one between each other is actually pretty similar. Yeah. And then of course there is also the crystal values for each of the group at the p value. So significant or not significant. Yeah. So I hope that you still remember about the statistical analysis. Yeah. Right. So that's for the evenness. So let's see for the faiths one. So the name is faith. Yeah, 40% box pop again. So for each of the body set, yeah, you need to look for the richness, yeah, to see this faith is actually for richness and based on the phylogenetic relationship as well. And you can see that the richness between the sites, yeah, it is differ pretty differently, especially like from gut when you compare with the palms, yeah, both palms, left and right palms, and also from the tongue. The one that have a lot of richness is the left and the right palm. Well, pretty obvious though, because yeah, this outside your hand. Right, and also the focus follow to see the significance with those. Okay. So already did this, right? The significance, and also the next one is for the beta. Yeah, for the beta. That's for the alpha for each of the area. For now, let's look for the differences between the community. Right, just copy paste this again. And then wait. Okay, let's take a look about the results. Yeah, it gives you the unvectored uniform, predict significant, and subject groups and significant as well. So, results, then did back based on body size. Let's see, based on body size. Okay, sample size, number of is 4, the p-value here, so that means they are significant, right? So based on the differences, yeah. So I think the one that will be the difference in the cut. And then yeah, so from distance to cut, yeah. So left palm to cut is significant. Right palm to left palm is not that significant. Tongue to another part is also not significant. And also some kind of another scientific analysis that you need to look at yourself. Okay. Okay, so then next is the this one is based on the body side. This one is based on the group on a subject. Yeah, so we only have two groups. You can see the p-value is much, so that means there are no significant difference between those two groups, right? Yeah, so yeah, so we can see that based on the microbial composition between one subject to another subject, yeah, it's not that much difference. Yeah, now you can create a lot of story. Okay. So next is creating the entropy plot for the from the PCOA, from the break artist, and also the unifuck. Yeah, since based on the day since experiment start. So copy paste again. Just okay. Let's take a pic again. Okay, the result is break artist and also unvectored. Start with the break artist. Okay, so this is based on barcode sequence. You don't really need that. Okay, so this is the based on the body side, based on the experiment. Yeah, so from left to right is right after the experiment until 168 days of the experiment. Look, this one is better. So this and we can delete some part of the data. Yeah. So the emperor is actually how you visualize them in some kind of figure. Sorry, graph. So can create story. Yeah. So then there's a question in that. So that is something that you need to answer. Yeah. Just think around the emperor plots. Yeah. And then check around how to create a PCOA, what PCOA means and something like that. So after that, you can create a story and then answer this particular question. Yeah. Okay. So, and then here the next part is also a rarefaction. This is based on the sampling depth. Yeah. To see how difference between the community between a different kind of the type of sampling itself. Yeah. Okay. Let's just copy paste this and let's let the software do the works. Okay. Into alpha fraction dot kzv, and it should be right there. There. So for looking for the view, we don't need to create a path to that because it is in our working directory, and just call it. So if side, yeah, you can see the features, and then the sunlessness, faithlessness as well for each party set. So you can see that from the tongue actually have a lot more richness in this particular area. More observed features in the tongue, both part and sorry, this one is left palm, right palm. The color is the same. Yeah, you can click this to open up. Oh, so that one is leg, right palm. Yeah, right palm. Yeah, right palm. So yeah, again, I believe it is pretty suggesting, pretty supporting, yeah, our previous data, so that both of the right and left palm to have more richness, yeah. And that is also have a lot of observed features, both of the indices from that pretty much. Yeah, refraction is just give you some idea. So how many markups are in there? Yeah. So far we do not yet identify it yet. So, but the reflection is looking for the unique sequences for each of the sample, which these unique sequences can be attributed to different kind of species. Yeah, usually can be attributed to different kind of species. Yeah. Okay. So next is now we're trying to look for the taxonomy analysis. Yeah. So this one is need to have a database. Yeah. So some kind of machine learning involved in this particular area, and we need to kind of like, in the simple way is that we have this a lot of sequences, and then we want to see whether it is actually identified using the reference from the Silva 16S classifier. Yeah, so this is something that we need to take a look around. So this one from the Chem2 already provided, yeah, for the feature classifier. Yeah, you can download via browser or using the WCAT. Here I'm just using the WCAT because I don't want to download from the browser. We already used the command line, so just use that faithfully. Okay, we need to download the database. Next is the feature classification. Yeah, feature classification using the sklearn. Yeah, copy paste this again to get the taxonomy classification artifacts, and also creating metadata tabulation. Yeah, metadata tabulation based on the metadata. Well, we need to wait. Okay, so we already have our taxonomy analysis, yeah, based on the 16S classifier from the Silva database. Let's look. Taxonomy GZV, I believe is in our working directory. Yeah, there you go. Yeah, based on this, yeah, because we from the comment didn't actually mention another part. Yeah. So we can pick it directly. Tools. Oops. Okay, so for each of the feature ID, yeah, feature ID, you can have identification. Yeah, so this one is the bacteria, Rickettsia, this is one Akkermansia municipalvalia. So this one is based on the taxonomic hierarchy, kingdom, phyla, class, order, family, until species, genus, species. Yeah. So this one we found Acidophorax. This one doesn't know until the species level, which is usually normal that we found in metagenomics, because again, 99% of the bacteria that at least we know of is not culturable in the lab yet. Yeah, so we can't really have a proper identification until species level unless we can have it in the lab, so we can replicate, see directly, maybe looking at the colony, look at the actual cell. Yeah, so because this is the one kind of challenges, looking for this particular field. Of course, for each of the feature ID and also for each of the taxon based on the sklearn and our database from Silva, and also the confidence of using that. Yeah. So the taxonomic table here maybe not going to give you a lot of information. Well, they give you information, but not in the way that we can see it clearly. The next one is we trying to create a power plot. Yeah, this is usually the visualization that we can give to in the paper or in the presentation later on, so that the other can see it clearly. Yeah, just see the power plot and then they can, oh, this kind of bacteria accessing this particular sample. Yeah, copy paste again, because they already provide the data. The data, the comment already. Yeah, and then let the magic to go. Oh, it's done. Oh, that was quick. Okay, taxa bar plots, KZ3, I believe this is still in the same place as our working directory. So let's view it again. Okay, so right, so for its sample here, and then this one, okay, all of them back here because we still are looking at the level 1 taxonomic level. Maybe look at this. Plot is just visualization, yeah. Okay, until level 2 of taxonomy level, we can have until phylum. Yeah, for example, maybe not going to be sort by sample. Can we change this? Yeah, we can sort this. Let's say based on policy. But we have more the purple one, synergy test, while this one have a little bit on the right one as well. So let's go deeper. Yeah, so now we're talking. Oh, what is that? Why? Oh, wait. Oh, what? Right. Sample. Okay. Yeah. So this is usually the one that we look at, and from there we can see that some actually are pretty dominant, especially in the cuts. Yeah, the green one. So bacteria data. Well, on your left palm, right palm is pretty diverse in here. Some actually have a very, very diverse area. Even though some kind of bacteria are also a little bit dominant. And then the tongue is the purple one. You can hover it to see what kind of bacteria there. Neisseria. I believe this one is responsible to gonorrhea, or at least perceptive. Yeah, definitely. Okay, tinker around to look for what kind of data that you want to take. Yeah, let's say maybe you want to change it with subject. Yeah, and look for what is subject for each subject here. Yeah, And then after that, this is the visualization that we expect, yeah, when reading some kind of metagenomic paper, and to see how the difference between, yeah, based on the question that the researcher wants to answer. Yeah. Okay, so the last part of the tutorial is creating what we call the NCOM. Yeah, NCOM. So this is looking for whether if one feature is actually abundant or present in different kind of abundance across the sampling group. So abundance testing. Yeah. Well, personally, I didn't use this a lot, but well, let's try it again. Yeah. Okay, copy paste again the comment that they are the generously provide, and then wait. So this is still creating a frequency table, a feature table, feature table. So we don't have a lot of visualization yet. So, and then this one needs a cut table, a feature table composition, another artifact. Okay, set up on it. Okay, so it's already creating the uncount subject visualization. We can pick it again. See what's information that we can get from that. Yeah. Okay, so this is what is the feature. Not a lot of things. This one is based on the subject. The feature that is actually exists in development subject. Well, you need to look at the documentation again about what this means. For now, we'll just work for the last part, yeah, I believe, for the composition, collapse, and also from the CTS. So, well, we found that some kind of feature actually abundance in different kind of subject at once, but we don't really know because the information is not really thing that we can tell about. So the last part is the put them using the taxonomy. Yeah, so now a little bit more readable. So that's one is just based on the sequence subject. Yeah, not a lot of things that we can work on. So this one on the table, copy paste this, and let the machine do the work. All right, that was quick. That was quick. So let's look again. Qiime tools view 16s and oh, that was L6. Oh, that's not 16, that's L6. Okay. Well, yeah, anyway. Okay, so that do not do anything for now. Let's say we can have now we have the name so that we can a little bit more idea of what kind of. So here we found out that the this one is actually accessing many kind of group at once, many kind of subject at once. Yeah. So yeah, we generate different abundance across the subject. Yeah, the subject it seems as more abundant. So this one is accessed in both subject. Yeah, from there we can need to look. Yeah, so maybe try to link it of what kind of backtraces and maybe the function, the non-function, or the known characteristics of this particular channel. Yeah, so I believe that's for the moving pictures tutorial. So I hope this give you a little bit of a light of what we working on it. So this one is pretty easy, and in the terms of looking for the typing in the comments or something like that, because they already put them here. So what you need to do is just looking for the results and interpreting the results. Yeah. So I hope that you get a lot of things from this, and don't stop learning. So yeah, working on this maybe at first is really hard to look at it because it's just lines or anything. Yeah, but again, when there's a will, there's always a way. Okay. So that is so. Thank you, and see you this Friday. Bye.